Welcome back to Clinical Hematology Reviews. Today, we'll be discussing peripheral blood smear preparation, or PBS preparation. Microscopic examination of the PBS remains the fundamental gold standard in diagnostic hematology.
It provides the critical morphological validation needed to confirm automated analyzer results. Over the course of this module, we'll break down the pre-analytical variables, physical mechanics of wedge smear preparation, Romanowski's staining chemistry, and troubleshooting protocols required for optimal diagnostic accuracy.
A peripheral blood smear is clinically indicated whenever an automated CBC analyzer flags an abnormality, such as suspected blasts, abnormal lymphocytes, or platelet clumps. It is also required for performing manual white blood cell or WBC differentials when instrument parameters fail or trigger laboratory review criteria.
Beyond confirming numerical data, direct microscopic evaluation allows us to assess specific red blood cell, white blood cell, and platelet morphologies. Recognizing pathognomonic shapes, cytoplasmic inclusions, and maturation abnormalities is vital for diagnosing anemias, leukemias, infections, and systemic disorders.
Diagnostic accuracy begins with specimen integrity. The standard anticoagulant for hematology is dipotassium EDTA, and smears must be prepared within 0 to 3 hours of collection when blood is kept at room temperature, between 18 and 25 degrees Celsius.
When storage exceeds 3 hours, significant cellular artifacts develop. Platelets swell and fragment, which can cause pseudo-thrombocytopenia, while lymphocytes and monocytes develop nuclear clefting, cytoplasmic vacuolization, and cloverleaf nuclear lobulation.
Neutrophils also demonstrate progressive chromatin pycnosis and detached nuclear lobes, while red blood cells deplete their ATP and form crenated echinocytes. Furthermore, underfilling collection tubes to less than 50% volume creates an excess concentration of hypertonic EDTA.
That hypertonic environment draws water out of erythrocytes, causing cellular shrinkage. This artificially decreases the mean corpuscular volume, or MCV, and falsely elevates the mean corpuscular hemoglobin concentration, or MCHC.
High-quality smear preparation requires clean, grease-free glass slides with beveled edges and frosted ends for labeling. Spreader slides must have smooth, polished, unchipped edges to prevent dragging large leukocytes unevenly across the glass.
Accurate dispensing requires transfer pipettes, or micropipettes, calibrated to deliver a consistent 5 to 10 microliter drop of well-mixed whole blood. We also require high-purity anhydrous absolute methanol of 99.8% purity or higher, polychromatic right stain, and a phosphate buffer maintained precisely between pH 6.4 and 6.7.
Macroscopically, an ideal wedge smear covers 2/3 to 3/4 of the total slide length. It should display a smooth, continuous rectangle-shaped outline with clearly visible lateral margins that do not touch the outer edges of the slide.
The terminal end must taper smoothly into a straight, square, translucent feathered edge without ridges, waves, or tails. When held up to the light, the smear should exhibit a uniform, translucent pink-purple sheen, indicating appropriate cellular distribution and thickness.
The manual push wedge technique begins by inverting the EDTA tube 8 to 10 times to ensure complete cell suspension. A 5 to 10 microliter drop of blood is placed approximately 1/2 inch from the frosted end, and the spreader slide is positioned in front of the drop at a 30 to 45 degree angle.
The spreader is drawn backward or forward depending on positional preference, smoothly into the blood drop, allowing the liquid to spread along the contact line by capillary action until it covers roughly 80% of the slide width. The operator then pulls or pushes the spreader away from the blood drop in a rapid, continuous single motion without altering downward pressure or lifting the slide.
Smear thickness depends heavily on blood viscosity, which is dictated by patient hematocrit. For severely anemic blood with a hematocrit below 30%, the blood is watery and tends to run off the end of the slide. To compensate, we increase the spreader angle to greater than 45 degrees and increase the push speed to create a shorter, thicker smear.
Conversely, for neonatal or polycythemic specimens with a hematocrit above 55%, the blood is highly viscous and produces crowded, excessively thick smears. In these cases, we decrease the spreader angle to less than 30 degrees and push at a slower, gentler speed to extend the film and establish a readable monolayer.
Technical errors during spreading produce characteristic macroscopic and microscopic defects. Hesitation or jerky forward motion causes washboard ridges where cells clump in waves, while a chipped spreader slide edge drags larger white blood cells into longitudinal streaks and tails, skewing the manual differential.
Placing too large a blood drop leaves no readable monolayer due to extensive cell overlap, whereas too small a drop produces an insufficient viewing area. Delaying the push stroke allows platelets to aggregate and red cells to form rouleaux, and applying excessive downward pressure crushes fragile leukocytes into unidentifiable smudge cells.
In cases of profound leukopenia, where the white blood cell count drops below 2.0 times 10 to the third per microliter, standard smears contain too few leukocytes for an accurate 100-cell differential. In this scenario, a concentrated Buffy coat preparation is required.
Anticoagulated blood is placed in a Wintrobe tube and centrifuged at 2,000 RPM, or 800 to 1,000 times gravity, for 5 to 10 minutes. The supernatant plasma is aspirated down to 1 millimeter above the cell layer, and the concentrated leukocyte Buffy coat is harvested to make a push wedge smear.
This concentrated preparation enables qualitative evaluation of white blood cell morphology, toxic changes, and circulating blasts. However, red blood cell morphology must never be reported from a Buffy coat smear because centrifugation causes artificial cell stacking, altered pallor, and severe distortion. Romanowski staining relies on a 2-dye polychromatic mixture dissolved in absolute anhydrous methanol. Methanol acts as a chemical fixative that rapidly dehydrates and binds cellular proteins to the glass slide without lysing cell membranes.
The stain contains basic cationic methylene blue and azure dyes, which carry a positive charge and bind to negatively charged cellular polyanions like DNA and ribosomal RNA. It also contains acidic anionic eosin Y, which carries a negative charge and binds to positively charged basic protein polycations, such as hemoglobin and eosinophil granules.
Ionization and selective dye binding are tightly controlled by the addition of a phosphate buffer maintained between pH 6.4 and 6.7, with an optimal target of 6.6.
If the pH falls below 6.4, excess hydrogen ions protonate cellular proteins, accelerating eosin binding and causing the smear to appear excessively red or orange with washed-out nuclei. If the pH exceeds 6.8, excess hydroxide ions accelerate methylene blue binding, causing the smear to become excessively dark blue with muddy slate-gray red blood cells.
Automated slide stainers standardize the staining process across 3 distinct stations. In Station 1, the air-dried slide is flooded with pure Wright's stain, allowing methanol fixation and initial dye contact to occur simultaneously.
In Station 2, the system dispenses the phosphate buffer using capillary action to thoroughly mix the buffer and stain to trigger controlled ionization. In Station 3, the slide is rinsed with deionized water to clear unbound precipitate, followed immediately by an integral air blower that flash dries the film to prevent water artifacts.
On a well-stained Romanowski smear, normal erythrocytes display a uniform salmon pink to pink buff color without gray or greenish tones. Neutrophil nuclei show dense, purple-violet condensed chromatin, while their cytoplasm displays a pale lilac hue filled with fine pink granules.
Lymphocytes exhibit rich purple nuclear chromatin surrounded by a clear robin's egg blue cytoplasm, while basophil granules appear dark purple-black and coarse. Eosinophil granules stand out as bright, spherical, refractive red-orange structures, and platelets appear as crisp violet-purple granular clusters.
Microscopic evaluation must be performed exclusively in the monolayer zone, also known as the zone of optimum morphology, located immediately adjacent to the feathered edge. Examining the thick body of the smear leads to false interpretations due to overlapping crowded red blood cells and distorted white cell sizes.
Under the 100x oil immersion objective, an ideal monolayer demonstrates erythrocytes evenly distributed in a single layer, barely touching edge to edge. Normal red cells in this zone exhibit a distinct central pallor occupying exactly 1/3 of the cell diameter, allowing crisp assessment of cellular inclusions and leukocyte chromatin patterns.
Systematic troubleshooting resolves common staining irregularities. Excessively acidic, bright mud smears with pale nuclei often result from a low buffer pH, overwashing, or aged stain where methanol has oxidized into formic acid. Correcting this requires recalibrating the buffer to pH 6.60 and replacing old stain packs.
Excessively alkaline dark blue smears are typically caused by a buffer pH above 6.8, prolonged staining time, unaged stain formulations, or inadequate rinsing. Adjusting the buffer downward, shortening staining time or extending the rinse cycle restores optimal color balance.
Two major physical artifacts frequently encountered are water artifact and stain precipitate. Water artifact manifests as refractile, glassy rings inside red blood cells, accompanied by artificial crenation caused by high humidity, water-contaminated methanol, or blowing breath onto wet smears.
Stain precipitate appears as amorphous dark blue-black particulate grit scattered across the slide background. This artifact results from using unfiltered stain solutions, allowing the stain to dry out during the incubation step, or providing an inadequate rinse volume.
When red cell inclusions are noted on a right-stained smear, confirmatory stains resolve diagnostic ambiguities. Prussian blue uses an acid ferrocyanide reaction to detect ferric iron, staining Pappenheimer bodies bright blue while leaving Howell-Jolly bodies and basophilic stippling negative.
Supravital stains, such as new methylene blue or brilliant crescel blue, are applied to living, unfixed erythrocytes. They precipitate ribosomal RNA and reticulocytes and reveal Heinz bodies, which are composed of denatured hemoglobin and are completely invisible on standard right-stained smears because methanol dissolves them.
Clinical quality control requires staining and reviewing a normal control blood slide daily to verify staining intensity, buffer balance, and cell morphology before reporting patient results.
Standard biosafety mandates universal precautions, including gloves, lab coats, and eye protection for handling whole blood. Methanol-based reagents must be used in well-ventilated areas away from ignition sources, and all glass slides and broken glass must be discarded immediately into rigid, puncture-resistant biohazard sharps containers.
To summarize our discussion, mastering peripheral blood smear preparation relies on understanding the physical mechanics of the push-wedge stroke, adjusting the spreader angle for patient hematocrit, maintaining strict pre-analytical timeframes, and controlling the chemical equilibrium of Romanovsky staining with a pH 6.6 phosphate buffer.
Today we covered the end-to-end technical and analytical principles of peripheral blood smear preparation, Romanovsky staining chemistry, artifact troubleshooting, and quality control.
Thank you for joining us, and we look forward to having you with us next time.